Great, okay, so I think we're good to go. Just can I have a quick show of hands? Hands up if you've ever made a cryo-EM grid before in your life, in your career. Fantastic. Hands up if you've crystallized something. Okay, awesome. So that was kind of what I was expecting. So we've got lots of structural biologists in the room, and I guess with the new technology that's coming into your institute, hopefully many more of you will be getting interested in single particle and cryo-Electron microscopy and also tomography workflows. So as I said, I'm going to talk to you about some of the work we've been doing in my facility team around particularly the single particle pipeline. So for the first section of this talk, everything that I'm going to be talking about has been done in collaboration with Steve Munch's group at the University of Leeds, and ably assisted by David, Yiheng and Izzo, who are fantastic PhD students in the lab. And this work has also been conducted in collaboration with Michelle Darrow at SPT LabTech, and I'll get into that in a minute. Okay, single particle cryo-Electron microscopy. What is it that we want to do? What do we want to achieve? So we want to obviously solve beautiful high-resolution structures of our macromolecules of interest. And in order to do that, we need to take our particles of interest, so this is just a schematic here, and have these particles suspended in a thin film of vitreous ice, where hopefully we're going to look at them in their native-like state and be able to solve their structure to a high resolution. So the first part of this talk is going to be really focusing on particle behaviour within this thin film environment and the things which might which impact on that. So we know from a whole series of studies. So for those of you who have made an electron microscopy grid before, when you made your cryo-EM grid, can you put your hands up if you've made a cryo-EM grid? We've got some hands, there's some more over here. Can you keep your hands up if the first grid looked great? So everyone's saying Misha's first grid looked great. So there you go, he's amazing at cryo. But pretty much every time we go through a single particle project and you put your sample onto a grid, almost for every single project, we require some kind of optimization because we're not getting this lovely schematic type situation here where our particles are lovely and monodisperse and adopting a wide range of conformations. We see a whole range of problems with our protein. Even when the biochemistry that we've done prior to making our grids all looks wonderful, we get the protein onto a cryo-EM grid and all of a sudden it doesn't look quite so good. Maybe there's aggregation, maybe we can't see our particles, and there are all sorts of things that can happen. So we started at this. So Steve Munch's group in Leeds is very interested in time-resolved electron microscopy. So this is developing technologies where we can make grids on a millisecond timescale and using that to study biological processes that happen within those timescales. But my real interest in this came in saying, can we possibly use this technology of plunge freezing? I'm just going to see if this video plays. There we go. So, we've got some aggressive sounds. Would someone mind muting Misha's laptop? There we go. I'll turn it off for a second. So, thanks, Misha. So, what we have here is a pair of tweezers which are holding our electron microscopy grid, and then just like on this schematic here, we have a nozzle that sprays out small microdroplets of our liquid containing our specimen of interest. It sprays onto the grid. So essentially the grid passes and it sprays onto the grid as the grid is plunging down, and then it plunges into the ethane. And by using this technology, we can make grids certainly within 10 milliseconds. So we can make grids really quite rapidly. So we came at this and started to ask, can we possibly use this technology to study how proteins are behaving in this thin film environment over time? And can we use that to design better strategies for our single particle cryo-electron microscopy? So yeah, thank you. Without the aggressive sounds now. So you can. This is a real-time video, so you can see this pneumatic arm is really plunging very quickly. So the other thing that we were interested in looking at is how sample preparation technologies, which aren't our standard filter paper-based, Vitrobot type technologies, impact. So we were lucky enough for this study to get access to the chameleon device, which is a next generation sample preparation device. They cost about £500,000, so they're not cheap infrastructure. And so we don't have one in my facility, but we went to go and use the development machine at SPT Labtech. So using both our traditional grid making techniques, our time-resolved spraying-based techniques, and this machine here, we thought if we do a study and we can compare the behaviour I guess, how proteins behave in this thin film environment throughout, then maybe we can learn something about how to optimise our sample preparation pipelines. So the first thing that I really wanted to show so before I go any further, I should say for this study as well, we picked three proteins, 3 different macromolecules of interest. So we had apoferritin, which is the cryo-Electron microscopist's favourite test specimen, HSBD1, which is a chaperone, and a ribosome. And the first thing we wanted to do is have a look at where the particles go to in the thin film environment. And we can see if we have a look at, so what we have here is on each of these graphs you have 2 layers. And on each of these, and the layers represent the thin film environment that's being formed, our layer of ice, and then the dots inside represent where our particles are partitioning to. So we can make grids on a Vitrobot, which is our traditional sample preparation technique, making grids very quickly with our time-resolved device, or making grids a little bit quicker, either with our a little bit more slowly, either with the time-resolved device or the chameleon. But really the take-home from this is that even when we make grids as quickly as we possibly can, almost all the molecules that we are looking at in our cryo-Electron microscopy grid are up at the air-water interface. So this air-water interface is actually a fascinating environment that's very, very different from the kind of environment that exists in bulk solution. So, you know, when we're purifying our proteins. And we know theoretically that our proteins can be exposed to forces at the air-water interface that might make them adopt non-native conformations or impact our downstream structure determination process. So the first thing we knew is that our proteins were up at this air-water interface. So what are they doing when they get up there? Well, the first thing that was really, really noticeable is that, so we know when we're working with our proteins of interest, our macromolecules, we understand, we can measure the concentration of our protein prior to it going onto a grid. And you can therefore theoretically calculate, if you know how thick your ice is, you should be able to predict how many particles that we should see in the ice layer as we're going through our cryo-Electron microscopy experiment. So we should be able to know how many particles we would expect. And one of the really interesting things that we see is that the number of particles in the thin film environment often doesn't correlate very well with what we expect. So if we have a look at the Vitrobot to start off with, so this is just your traditional technique for preparing your grids, we can see compared to this red line here, which is our predicted how many particles we would expect to find in the thin film environment, we actually get a huge concentration increase with our protein. So essentially our protein is concentrating up in this thin film environment that's being formed. And if we use our alternative, there we go. If we use our alternative sample preparation methods, so the time-resolved device or the chameleon, for example, we don't see we sometimes see a little bit of a concentration effect, or we don't see a high concentration effect at all. And this is really fascinating to me because as somebody that works in a cryo-Electron microscopy facility, one of the most common questions that I get when people first come to the facility is how much protein do I need and what concentration does it need to be at, and how pure does it need to be? And it's always very frustrating for me because I can never give a clear answer to this. I can never say, oh, well, you need this exact amount of protein, because the proteins in anecdotal evidence will concentrate up in different ways on the grid. So this is really, I guess, one of the first slightly more, I guess we went through a process where we measured this and did quite controlled experiments to be able to look through the protein concentration. and really demonstrate that actually, if you look across the board here at these 3 different test specimens, they all concentrate up in different techniques, but they do so to differing degrees. And this helps to explain the observation that we have, that actually it's very difficult to predict how much protein you will necessarily need. But one thing is for sure, if you're using these traditional filter paper-based techniques, making your grids on the Vitrobot, then you will be experiencing a concentration of your sample, which is actually great because it means that you don't have to prepare as much protein. And if you're going to some of these other sample preparation techniques, you will know right from the get-go that you're going to need a higher concentration of protein. Okay, so the proteins are going up to the air-water interface. Once they're up at the air-water interface, they're often being concentrated. So we're seeing that interface acting as almost a sink for our particles. But what do they actually do when they get there? Yes?
Is there no comprehensive study on what fraction of protein goes into the water interface through the, crossing the air-water interface? Or is there any physical property of protein which correlates at all with this partitioning?
Yeah, it's a great question, and the answer is it doesn't seem to be particularly straightforward. So just in the same way, yeah, it's a really good question. Do you mind if I'll present this slide, if that's okay, and then I will come back to it, because it's a really fascinating question, and that really gets to the heart of what we're trying to achieve. Because if we can understand protein behaviour in the thin film sufficiently, we can get to the point where we're not guessing anymore, and we can actually make good decisions before we go into this. So once the proteins are up at the air-water interface, what happens to them? So if you think about your protein, your macromolecule of interest, that's a really interesting chemical landscape across the surface of your macromolecule of interest in terms of charge, hydrophobic, hydrophilic interactions. And we were really interested, and we know that one major problem in cryo-electron microscopy that many projects encounter is what we call preferred orientation. So if we think back to that schematic right at the beginning where we had our particles nicely distributed in the thin film environment, those particles were randomly distributed. And that's really required if we want to solve a high resolution structure by single particle cryo-electron microscopy. But what we often see is that our protein of interest, for example, this slide changer, rather than adopting lots of nice random views, will stick up at the air-water interface in a preferred orientation, which then makes high-resolution structure determination very challenging. So we looked at this, we had a ribosome sample, and the ribosome sample had actually fallen, it was a 70S sample, but it had also fallen apart into 50S and 30S. So what we did is so this is data all from a single grid, and we, again, made grids at different time points, so 13, 54, 200 milliseconds, and then our seconds-long Vitrobots. And we looked at the angular orientation of our particles present at each stage. And really interestingly, we see that for the 30S and the 50S ribosome, where you see these hot spots here, this indicates that we have essentially a strong preferred orientation, and when we see more of this diffuse red colour, that means we have more of an angular spread. So over time we can see more of these hotspots developing here, and again here we can see more of these hotspots appearing. So basically the longer it takes us to make our grids, for these two samples, the worse our angular orientation is getting. But the really fascinating thing is that for the 70S ribosome, actually it goes up at the air-water interface, adopts a really strong preferred orientation, and then over time it then seems to diffuse out a little bit and we get more angles over time. So this is another piece of evidence that's showing us that really these are all subsets of the same macromolecule, but actually we're seeing quite different behaviours with them across the piece. And I think that these data get to the heart of why single particle cryo-electron microscopy pipelines can be so challenging to optimize, because we do see a great deal of project to project variability. So yeah, once the particles are up at the air-water interface, they are potentially adopting a preferred orientation, which may get better or worse over time, depending on the sample. We've now done this kind of analysis on a range of different proteins, and generally your preferred orientation does get worse over time. We have fewer examples that it gets better over time. So is that depends on the type of the grid you use, or irrespective of the grid you use? So great question. So this is all data collected on our 1.2, 1.3 quantifoils, so just standard carbon, not gold mesh or anything. But I will come back to grid technology a little bit later. So the next question for us really was thinking about how these proteins are behaving up at the air-water interface. So we've already said the air-water interface on the thin film environment formed during cryo-EM sample preparation is a very different environment to that formed during in bulk solution. And the main driver for that, the main reason, is that we have an environment where our protein can potentially start to unfold and be able to satisfy the parts of the protein that like hydrophobic interactions and the parts of the protein that like hydrophilic interactions. And those forces present at the air-water interface can induce an unfolding in our protein. So we started to wonder what was actually happening to these molecules over time and whether we could see any, for example, local unfolding or disorder starting to come out in our proteins. So we did this, again, looking at the ribosome sample. And if we had a look at the L31 subunit on 70S, we can see we see a little bit of we see some density for this at 13 milliseconds and 54 milliseconds, but then by 200 milliseconds and 6 seconds, that's really dropping right off. I think this is an even on the 50S ribosome, I think it's even clearer. So again, 13 milliseconds, 54 milliseconds, we're seeing nice density for this part of the subunit. But by 200 milliseconds and 6 milliseconds, we've lost that density. So that's telling us on a timescale of hundreds of milliseconds, we're starting to either have local unfolding or disorder generating in these regions, which if you're interested in L31 would be extremely annoying for your study. And then a final example here, something which was a little bit slower. So the S2 subunit here, we see really strong density at 13, 54 and 200 milliseconds, but 6 seconds on the Vitrobot, we see no density in this region. So I think if we go back to, yes.
Sorry, I mean just a naive or practical question basically. So suppose say your protein doesn't go into the water interface.
Yeah.
What are the different methods, I mean, like you scientifically doubts to actually bring it onto the grid?
Yeah. So the good news is that there are lots and lots of experimental approaches that you can use. If your protein is up at the air-water interface, there are lots of experimental approaches that you can use in order to get rid of that problem. The first one is to sequester your protein away from the air-water interface, so to use a continuous support, graphene oxide or continuous carbon, for example. Another one would be to modify the properties of the air-water interface itself, so adding a surfactant onto your grid, exactly, which absolutely works as a strategy, and then there are now lots of examples out there. But going back to your point around, can we, exactly, yeah, but it's very, very difficult to, modifying the interface in such a way that you start to see a range of orientations is something that requires project to project or a variation. So again, there isn't just a single detergent at a single concentration that seems to work.
Changes in the amino acid constitution of the protein itself.
Yeah, absolutely, absolutely. And I think there are almost certainly buffer composition will, in terms of salt concentration as well, any other, anything else which impacts viscosity. Essentially anything which impacts your fluid dynamics of your thin film formation or then the air-water interface itself is going to be potentially impacting.
So in other words, if I can predict actually the air-water interface interaction of the protein,
yeah,
I should be able to predict actually what will go inside.
Yeah, so the absolutely, and I think that's one of the so there are a couple of different approaches here. One of them is if we can understand those air-water interface interactions better, then we can start to design more generic strategies around it, which would be great. The second one is that you just keep your protein away from the air-water interface. So you design other technologies that allow you to do this. And like I say, continuous support films are a really popular approach. So like I say, graphene oxide, graphene, other engineered supports. But in addition, there are some really cool projects looking to develop microchips. So you would have a chip with a tube that you would then put your sample into via kind of capillary action, and that sample will have never experienced any kind of air-water interface. Exactly. I mean, we may end up with other issues around flow-based aggregation, for example, and shear forces. But you know, these are all technologies that I think are really exciting. So So I've kind of gone through some of that initial, I guess, so we published that work a couple of years ago now, and there have been other fantastic papers from others in the field talking around protein behaviour in that thin film environment. So I wanted to present now some of our most recent work in this space. So while we were going through this study, David, who's a very talented PhD student that's just finished from Leeds, was model building into this HSBD1 data set, and he was doing this for the Vitrobot blotted grids. And he was model building, and he happened to, when he was building the cysteine in at this particular location, he just happened to notice that there was a suspicious little bit of extra density here. So he thought, oh, okay, this is interesting. And he brought it to the meeting, and we had a little bit of a discussion around it. And it became clear that this cysteine was actually in this location, was actually also associated with this loop region, which in David's electron microscopy EM density was disordered. So we weren't seeing any density for this particular loop region, but we knew from other structures that existed and had been published that this loop region was normally ordered in cryo-electron microscopy. So we thought, in cryo-EM data. So we thought, oh, this is very interesting. What's going on here? What is this mysterious additional density? So luckily, because of all of the previous work that we've done, we had a map available. Where we'd made grids very quickly, 14 milliseconds. And when we looked at the 14 millisecond grid, we saw the cysteine didn't have an additional funny density, and we had a perfectly ordered loop region. And we also happen to have a Vitrobot grid. We've solved a lot of HSBD1 structures at this point. So we also happen to have one where we'd added DTT into the buffer. And whereas in the previous ones there wasn't any reducing agent. And lo and behold, once again, we saw that there was no modification on the cysteine, and we had this beautiful ordered loop region. So this immediately got us thinking, maybe this suspicious additional density that we're seeing here is a product of oxidation. So we thought this was very interesting, but it's a funny-looking density on a random cysteine that probably is. Maybe this is just something that's unique with this map. So the next thing that we did is Yi Heng, who's a PhD student with Steve and I, went and did some searches of the EMDB just to see if we could find any other examples of any other protein systems that seemed to have this interesting modification. So if we have a look at beta-galactosidase, which is probably the cryo-electron microscopist's second favourite test specimen, again, there's lots out there on the EMDB. We can see if we have a look at this structure here, which has TSEP in the buffer, again, we have this additional suspicious density here. And if we have a look at a map with 5 millimolar DTT added, then again, we have no density. So we immediately thought, oh, well, this is very interesting. But of course, we all know that maybe materials and methods and published data is hopefully always accurate, but we often want to repeat this in our own hands. So we took a beta-galactosidase and we did a series of controlled experiments in our hands. So I'll just put these up. So again, we made a Vitrobot blotted grid, and for this particular cysteine residue on beta-galactosidase, we saw an additional extra density. If we make grids quickly, we had no additional density, and if we added in DTT into our sample, again, we have no additional density here. And I think one of the most interesting things about this structure is that if you have a look at this histidine down here, you'll see that we've got a change in rotamer location position between the blotted grid, where it's in this location, and then where we don't have the modification, it's changed its rotamer position. So this is a subtle structural change, but where these modifications are existing, we are seeing subtle but potentially important changes to our protein structure, whether it's rotamer location or whether it's order of a loop region. So the next question for us really is this something that's happening at the air-water interface, or is this just something happening within the thin film environment somehow? So we this time made a standard Vitrobot blotted grid, but this time with a continuous support film. So we just used carbon in this particular case, so sequestering our particles down at the air-water interface, and lo and behold, we have no cysteine modification. So we're confident that this is a process that's happening very close to or at the air-water interface. And I guess the final interesting observation that came out of this section of the work was that where the modification was existing, so for example on this blotted beta-gal grid where we have no DTT in the buffer, we noticed that our particle distribution was much less monodisperse than when we add in DTT into the buffer, and we end up with a much more monodisperse distribution of particles. So this seems to be having an impact. The presence of the modification seems to be having an impact on protein distribution within the thin film environment. So we had a question about grid types earlier. So as I say, pretty much all of the work we've done so far has been on quantifoil 1.2, 1.3s. But we had good anecdotal evidence that when you use ultrafoil grids, so these are grids where the bars and the mesh itself is all made from gold, rather than having, for example, a copper mesh and then a gold support. So we had some good anecdotal evidence that proteins are often seem to be more monodisperse if you use an ultrafoil grid compared to a standard copper grid. So we thought, well, maybe there's a relationship between this modification that we're seeing on the cysteine. And I'm going to run through this quite quickly, but the answer is basically if we make an ultrafoil grid, again, we don't see this modification. And we wanted to know whether this was really related to the copper or the carbon on the standard 1.2, 1.3 grids. Basically what we can see is that we don't get, if we make a gold carbon grid, we don't have this modification. So it's really only when we're using a copper grid do we get this additional modification. So we think that this is basically copper catalyzed chemistry. So I guess to sum up this little section of the talk, I think we've been able to show that oxidation of cysteines occurs within the thin film environment during during single particle grid preparation, and this can result in subtle but potentially significant structural changes. The good news is that there are loads of ways that this cannot be a problem for you. So you don't have to go and use a £500,000 machine if you want to fix this problem. Just adding DTT into your buffer, making grids quickly works, sequestering your particles away from the air-water interface, all prevent this modification from forming and therefore the downstream impacts on your protein distribution, for example. We're pretty confident now that this is copper catalyzed chemistry, but we're processing some data at the moment with, for us to really start to pin that down. And one thing that I haven't really spoken about so far, I've been showing you results for specific cysteine residues where we seem to see this impact. We haven't seen this modification apart from on surface exposed cysteines, but we don't see it on all cysteines all of the time. So there clearly is an impact around local environment there, which is interesting. And we've also seen other residues become modified at this point. So really, there's some really interesting things going on here. So the next thing is a little bit of a plea to you all. If you are model building into your cryo-Electron microscopy maps, into your EM density, and you see a funny looking cysteine, please do drop me an email and let me know, because we're trying to collect examples of this, and I think we've got about 20 so far from people who have seen this and either have they either have a with and without structure or sometimes just with, but I'd love to hear from you if you've experienced this. So what's the underlying mechanism on this? The short answer is we don't know, but our current working hypothesis is that the electrical field which is present at the air-water interface may be sufficient to split water molecules into hydroxyl radicals, and this combined with the copper is really driving the modification on the cysteine that we're seeing. But as I say, there's some interesting chemistry here that we're still trying to work through. So to sum up this section of the talk, I guess the take home for me is that we try as structural biologists to solve native-like structures of our proteins. Obviously, we're ripping them out of their native environment in terms of their cellular environment, either purifying them to a greater or lesser extent and trying to solve their, and then putting them into this thin film environment. So this clearly works as a strategy. There are many fantastic single particle cryo-electron microscopy structures that have been solved. But I think it is worth, as we're going through our projects and as we're looking at designing our projects, to bear in mind all of the different things that can potentially happen to our proteins within this very unique environment that our protein's not really ever seen before it gets there. Understanding a little bit more about this will hopefully allow us in the future to design better strategies. I've got to say that what we really wanted to do was design these generic strategies to say, hey, these are the things which will generically help. And so far, our work to date has really only shown that there is a huge amount of variation on a protein by protein basis, which at least, I guess, for me, provides some comfort when I'm struggling to optimize a project, because it is a protein-to-protein specific thing in terms of its behavior, but there are lots of strategies that we can use to improve the prep. Okay, so that's to sum up the first section of the talk, which was, I guess, more so focused on the sample preparation. And the thing I want to run through next is really thinking about now the future of cryo-electron microscopy and where things are going, because I think this is such exciting technology. And we had the resolution revolution in terms when we started to get Titan cryosys being installed, when we had the new direct electron detector technology go in. And I think it's tempting, and I guess, you know, well, Misha, you were telling me you collected on a cryos when, in 2009, yeah. So these cryosys have been in now for a number of years. And I guess they really started to become more widely adopted in 2014, 15, and we're now seeing an expansion in this infrastructure globally, which is just incredibly exciting. And I think it's tempting to say, like, oh, well, we've had, you know, massive improvements in terms of the microscope hardware, in terms of the detectors. You know, from here on out, all of the exciting stuff is happening outside of the microscope itself. But actually, we've really seen, even in the last couple of years, again, new technology is starting to come online. And these include something called aberration-free image shift collection, or that's what Thermo Fisher calls it. So basically, we have the ability to collect data rather than moving our microscope stage around all the time when we want to collect pictures. We can just micrographs, we can move to a single location and tilt the beam in order to collect. So basically, we have to do fewer stage moves, speeds things up, you get more data. Fringe-free imaging, which is a hardware modification on your microscope, where it means you can basically get a smaller illuminated area around where you want to take a picture, which means you can take more pictures in a particular region, you can take more micrographs in a particular region. We've had new generations of direct electron detector come online, so Falcon 4, Falcon 4i, and also new energy filters in Selectris. And we've been lucky enough at Leeds to put all of these things in place within the last year. And as a facility focused person who's looking to support researchers through these pipelines, we wanted to ask, what are the impacts on single particle cryo-EM pipelines? So for this section of the talk, all of the hard work was done by Yehuda, Josh, and Louis, who are in my team at Leeds. So what we did is we took this protein VIP3A, because if I do another apoferritin structure, I'm going to go crazy. So we wanted to pick a different protein, and we picked it because it's a very tractable target. It's a soluble protein. It's relatively big, 360 kilodaltons. It's a little bit symmetric. It's got C2 symmetry, but it's not the easiest protein in the world. It's quite aggregation prone. It adopts a preferred orientation. So it's not like apoferritin or something where you can get to an amazing resolution with one single micrograph of data or something. So we took a single grid of this protein, and we took multiple 4-hour data collections using a whole variety of different hardware and software. And what we really wanted to do was compare the performance of the different detectors. So we collected this 4 hours of data, and we put it through the same image processing pipeline in order to generate our structures. And here are just 4 shown on the screen here. So we used the Falcon 3 in linear and counting mode, which I won't go into detail. The Falcon 4, which is the new detector from Thermo Fisher, and then also in combination with the Selectris energy filter. So essentially, when we look at the resolution of the maps that were achieved from that 4 hours of data collection, we can see that the Falcon 3, which was our older detector, the resolution of these maps was poorer than the Falcon 4 data, and we saw that the Falcon 4 Selectris was even more improved again. And if you have so we can have a look at global resolution, and we also see the B factors that were applied. We see a trend in that as well. So this is really indicating that the quality of these reconstructions from the Falcon 4 are genuinely are a better quality reconstruction. So that's excellent, because we've just spent £1.5 million with Thermo Fisher, so it's excellent news that the new detectors are indeed doing their job, and we're getting improved resolution structures out of them. But especially if we compare the Falcon 3 counting dataset, in 4 hours we were only able to collect 340 movies, whereas in the Falcon 4 data we were able to collect closer to 1,500. So the amount of data in these reconstructions is very different. So we went through and we selected 10,000 random good particles from these data sets, And again, we still see this positive trend in terms of the resolution. So with the Falcon 4 outperforming the Falcon 3, and again, we see this trend in B factors. And the thing that I guess I wanted to highlight with this is that for a non-trivial biological specimen, this is the equivalent of taking 16 minutes of data acquisition to solve a 3.5 angstrom structure. So, you know, quite often in the facility, our standard data acquisition time is 24 hours. So we're allowing people to collect data for 24 hours, and then they'll go away and process that data. But actually, for many of our biological questions, 3.5 angstroms is a resolution that's more than sufficient for us to tell whether our fab is bound, for example, or to make an assessment as to whether our subunit of interest is present. And I just think this is absolutely amazing that we're able to get such high quality reconstructions with such a small amount of data from these new detectors. So the next question for us was really, what impact does the Selectris energy filter also have? So Misha's going to present some tomography data in the next talk, and for electron tomography applications, an energy filter really absolutely gives you an advantage because we're tilting our specimen, we have thicker specimens, and it helps us to collect data with better signal to noise, basically. But for single particle cryo-electron microscopy, the case has always been a little bit more nuanced as to really whether the energy filter provides you with any benefit or not. So we went through and collected 3 data sets, one with no no slit in. So this slit width here, so we've got 10 EV and 5 EV here, basically means you're tuning the filter to get rid of all electrons that have lost a particular energy. So in this particular case, we ran with 10 EV and 5 EV. We saw actually pretty clearly that using the Selectris with a slit, again, we're seeing an improved resolution from this in terms of the 4 hours of data acquisition. There are some really, and again, we see this same trend in terms of B factors. So I don't have time today to go into the reasons behind this, but I think that this is really interesting and certainly for me was slightly unexpected. But I'd be more than happy to talk about that in more detail later. So I guess takeaways from this section, the new detector technology coming out, particularly like I say the Falcon 4 that we've just had installed in Leeds, is just amazingly fast, and I think it presents us with a whole set of new challenges in terms of how we make the most of our cryo-electron microscopy technology, how we make the most of the infrastructure that we have available to us. Because if you can answer your biological question in 15 minutes, it's much more efficient for you to do that than for you to collect 24 hours of data, have to take that data, move it, use computational resource to process it, and then depending on your funding agency, you may have to keep that data for 10 years, for example. So, I think we really need to think carefully, and I think working within a cryo-electron microscopy facility at the moment is fascinating because we need to work out a way of getting the most out of this incredible technology that we now have at our disposal, whilst also ensuring that people who work within facilities can sleep. So yeah, it's really interesting. The one thing I will say, although like I say, we've been super impressed with the Falcon 4 Selectris being installed at Leeds. Actually, I've never come across, we've had hundreds and hundreds and hundreds of projects through the facility, and whereas a different type of detector or a newer detector might help you to push the resolution slightly, it might help you to get to the same place faster, but I've never had a scenario where it takes a project that was just intractable and wasn't working to one where to one where you could then solve the structure. So although I think it is worth us thinking about our detector choice and our data acquisition parameters, depending on the hardware that you have available to you, you don't necessarily have to get you can make up for it in other ways if you don't have access to the latest detectors. Okay, so just as a final couple of slides, I guess I just wanted to introduce the facility that I look after. So I work at the University of Leeds and I head up the Astbury Biostructure Laboratory facility. So we're extremely fortunate to have 2 Titan Cryoses. These were installed back in 2016 now. And we have 3 other TEMs and lots of other kit. And we really work with a whole range of users, everyone from looking at cells embedded in plastic right through to high resolution structural biology, in situ structural biology, and correlative imaging. And I've got to say that probably the most challenging and the favourite year of my life was when we had the cryoses first installed in Leeds. It takes so much to get this infrastructure in and to get it working. It takes so much at every level from everybody who's involved in these projects. When they're in and then everything's working and you realise that all of your hard work is paying off and you're able to do the most amazing stuff that you weren't able to do before, it's fantastic. So I'm super, super excited for you that you've got this, that you've got the cryos coming in, and I'm just so excited to see all of the amazing science that's going to be coming out when you've got this infrastructure available to you. So one of the things I'm quite keen on is sharing of best practices and information. So at Leeds we run lots and lots of training courses, and most of these are available to access remotely and virtually. So please keep your eye out on those, and if anyone has any questions about training, please do feel free to drop me a line. We also offer access through iNext Discovery as well, but like I say, you will have your own amazing infrastructure available locally, so you won't need to do that. Okay, so that's all I've got to say. I'm on Twitter, please tweet me. This is my fantastic team within the electron microscopy facility. So Louis, Tom, Oksana, Yehuda, Josh, Martin and Emma. And I have the great benefit of working with, so last year in the facility we had 130 users coming through that we provided training to, and we supported their projects, and that was just internally. So I'm immensely fortunate to work with this fantastic bunch of people, yeah, they keep everything fun. Okay, that's all I've got to say, and I'll be happy to take any questions.
Any questions?
So one of the questions you said in grid preparation that we don't know how those things. If we put some polylysine or something coating on the grids so that they take only one kind of this one, what happens?
Yeah, it's a great question. So in terms of your polyl-lysine, you can do that in a couple of ways. So you can have a continuous support that you treat with polyl-lysine, and that will absolutely just help to stick the particles and absorb the particles down. Sometimes it can induce a preferred orientation, sometimes it can make it better actually. Quite often your polyl-lysine will make it better. The problem is that you've then introduced a layer of additional noise into your images. So it's an additional strategy to still have a hole in your grids, but to have polyl-lysine coating on the carbon around, and that there is some evidence for some projects that even that can impact your protein orientation within the thin film environment, possibly because some of the polyl-lysine has come off and is acting as a surfactant at the air-water interface. But again, when we're thinking about ways of modifying our protein behaviour within the thin film environment or on the grid, we're always doing a bit of a trade-off, because it's better to solve a structure than it is to not solve a structure. So I always say if there is a strategy available to you that allows you to make progress, even if it's not ideal because you have polyl-lysine and it's adding noise, or even because you're adding a surfactant and that might have impacts on protein stability, sometimes it's still worth exploring even though it's not ideal. But most of the strategies that we have available to us introduce another problem. So we're always constantly having to trade off these different strategies with each other.
Rebecca, really quickly, I guess you answered this already, but how does the bioquantum K3 compare directly to the spectrus? And you've done any side-by-side comparison?
Yeah, so we specifically avoided doing side-by-side comparisons so that we don't get sued by one or both manufacturers. No, so we don't have access to, we had a bioquantum K2 in Leeds, and the plan was to do a K2 analysis, but when they installed the Falcon 4, they broke it. So we weren't able to do that experiment. There is some so facilities generally tend to be very nervous about wanting to say one detector technology is better than another. At least in part because, as I guess I alluded to, for most projects it won't be the thing that makes or breaks your project. It's really talking about pushing the boundaries. But I think the Biocontinuum K3, there is certainly evidence that's not published, but that I've seen that, again, it still seems to improve the single particle reconstructions. That becomes less surprising when you have a less optimal sample and ice is a little bit thicker.
Right.
And that was that particular case. So yeah, but it's interesting.
Any questions from the students? You can take one or two questions. No, everybody's overwhelmed.
Yeah.
I'm really glad for this talk because, you know, we always think that, you know, crystallography, you have to crystallize something, and cryo-EM is so much easier. And then when we start doing it, you realize there is so much work goes into sample preparation and optimization. So I'm really glad that you discussed all this in detail. Thank you very much.
Thank you. But like I said, the good news is there's lots of things you can do, so it's all good.
Okay, so the next speaker is Misha. Yes. And he's a investigator at Max Bilbrook Center for Molecular Medicine in Berlin, and his laboratory primarily works on membrane proteins and ion channel proteins. So they do both single particle reconstruction and in situ structure.